Okay, the next part of this chapter is to cover GMOs, and we're going to start just by describing two different approaches to modify traits of crops and animals. So one approach is just referred to as selective breeding, and this is something that has been done for tens of thousands of years. And so the idea behind selective breeding is that you don't really know anything about DNA. All you know about is the phenotypes of the traits you like. And so if you select and choose the animals or the crops that have the traits that you like, you naturally create evolutionary pressure in order to change some sort of feature of the organism. So if you've ever seen pictures of corn, ancestral corn, what it looked like before humans have bred it, you know, it's a much smaller seed. It produces much slower. It produces less caloric. The calorie content is much lower. So there's all sorts of things about the selective process that actually allows us to really improve the generation of crops. And this is a technique that has been used by farmers for generations and generations. Nowadays, we actually can take a completely different approach. So instead of worrying about the phenotype of the corn, we can start to worry about the genotype of the corn. And so this is where we choose to modify organisms because we want to incorporate specific genetic sequence into their genome. And so this is becoming more and more popular. It includes many different types of transgenic crop plants. Alfalfa, corn, rice, potatoes, tomatoes, tobacco, wheat, and cranberries. These are all examples of crops that have been successfully modified so that their genome is no longer the same. So selective breeding, so corn, ancestral corn versus modern day corn. The ear is much bigger. The kernels are much larger. The kernels have much more calories within them. The corn grows much faster, and this is all just phenotypic based. Selection on chickens, is equally as powerful. You can also choose, you can also select on chicken size, for example, and how fast they grow. So this image right here is just a comparison of chickens from 1957, 1978, and 2005. And the reason that these animals grow much bigger is just due to selective breeding. We have chosen the animals that are bigger, and we allow them to mate with each other. And so over time, you select for the genetic factors that cause the traits that you want. So again, it takes advantage of existing genetic variation, and kind of essentially what it does is it makes rare genetic variation common in the population. So let's say you have a population of dogs that you want to select for a very particular coat color. So let's say you wanted to look for brown spots on the dog. And so in general, if you have some sort of breed and you actually look at all the hundreds of thousands of dogs that are of this breed around the world, the idea is that some subset of these will have the trait that you're looking for. So maybe 1 in 100,000 of the animals has that trait that you want. But now, if we identify all the individuals with that, so 3 different individuals that have that trait, and we choose to use them to propagate the next generation. So, this individual, and this individual is not allowed to reproduce. Then what we're doing is we're artificially increasing the genotypes that are present within these dogs. And so if we allow these to breed together, eventually what happens is that the majority of the dogs will start to gain that trait. But we have no knowledge. What's important is that we have no knowledge of what the genes are that cause the trait that we're interested in. So we don't know why these dogs have this color. In general, we have no idea what gene causes it. If I gave you the DNA sequence, if I told you this is the DNA sequence of the dog, can you predict what the color is? I'd have no way to do this. So this is where I say you're blind to the genotype. You have no idea what the genotype of the dog is. All you know is the phenotype of the dog, and that's how you use selective breeding, is that you only care about the phenotypes that you care about. But now we have the opportunity to use GMOs. And so now we're choosing how to modify the DNA. So we can delete genes, we can add genes, or we can modify genes. So we talked in chapter 20 about manipulating mice. So what if we do this to crops and other animals that are important food sources? So now we can artificially add traits that make food production better. So we can force animals to grow faster, grow bigger, have a higher yield, increase nutrition, but also we often make it easier to grow them. So the unfortunate nature of factory farming in the U.S. is that we have very high population of animals that are grown together, and so these animals are much more susceptible to diseases. The higher dense, the more animals that are around, the easier it is for disease and pests to spread. So this could either be pathogens for cows and chickens, or it can be insects that eat corn or some other sort of crop. And so we can engineer these animals to become resistant to those diseases, pests, and insecticides. We can also engineer animals to be more tolerant to stress, and this is obviously important. It's global in the context of global warming. So as climate is changing in many places, the traditional places that we grow crops, the average temperature is getting hotter. And so we can now start to engineer plants to be more tolerant to changes in climate, either in hot heat or potentially cold or drought, so lack of water. And so this is also powerful to be able to expand where we can grow crops at. An example of this is in salmon, the AquAdvantage salmon. So this is a salmon that has been genetically modified in order to grow bigger and to grow faster. So this is an example of the salmon with the genetically modifications to it. And this is just in comparison to its wild type companion. So clearly this is able to grow much faster. And so the way they achieve this, one of the ways to achieve this is through selective breeding, where you just constantly select for the salmon that grow the largest and allow those to breed together. But instead what they did is they found two important genes that were identified from other fish. So these are completely different fish species. So one of them is the Chinook salmon and the other is the ocean pout. So one of these contains a gene that causes the animals to grow faster, and another one contains a gene that allows the fish to survive in colder environments. It's an anti-freeze protein. And so when you combine those genes and add them to the Atlantic salmon, you're taking DNA from a completely different species. It's still able to work and do the same sort of thing in the Atlantic salmon. And so these GMO salmon grow much bigger and are actually much more resistant. Another example is transgenic cows that fight injections. Mastitis, I think it's mastitis. And so mastitis is an infection of the udders, where this bacteria, Staphylococcus, is able to infect parts of the udder. And so these cow have been genetically modified, so they're now transgenic cows, which means that they contain a foreign gene that's not normally within their genome, and they were generated that possessed a particular gene, the lysostaffin gene, and this is a enzyme that actually cleaves the cell wall of these bacteria. So by releasing these enzymes, any of the bacteria that make their way that infect the animals, they're actually destroyed. They're actually destroyed by the emission of this protein. And then finally, in the pet industry, people have engineered zebrafish. So zebrafish are a very common model organism that's used in the lab. So experimenters have developed a lot of techniques for transgenic modification, and so they've been able to add different colored fluorescent proteins, green, red, or orange. And so, it makes it more attractive. People love brightly colored fish for the aquarium. And so it's just a way to engineer fish to be more visually appealing to human buyers of the marks of the fish. And then finally, we'll talk about mosquitoes. There has also been an engineering in order to control mosquito populations. Particular this species, Aegypti, and the approach is to actually manipulate male mosquitoes. So male mosquitoes do not bite or spread disease, so they do not actually suck on blood. They do not feed on blood, but these mosquitoes mate with females that do. And so they were able to genetically modify these mosquitoes to carry a lethal gene within them. And so the idea behind it is that if this mosquito mates with a wild type female, the offsprings will all die. And so large amounts of these male mosquitoes are produced and released into the wild, so that the most likely male that a female mosquito encounters is one of the genetically modified mosquitoes. And so, the most likely pairing is between a wild type female and this modified male mosquito, and that results in the majority of the larvae actually killing. So it's just a way to try to control mosquito populations.